Hello everybody, welcome you to our fourth and final hangout of the MOOC on HIV science. I'm Hilde, your community manager, and here we have both MOOC directors.
Hello, I'm Michaela Müller-Tutwin. I'm one of the two directors, and I welcome you to the fourth hangout and hope you have questions.
I'm Maciej Sathirion. I'm the other MOOC director.
So we are very proud actually that we had in total 4,424 students enrolled from 131 countries all over the globe, with a focus of course on France, Northern Africa like Morocco, Tunisia, but also Cameroon, Algeria, the US, Europe, South Africa, Cote d'Ivoire. So we had many countries present and many more, and we are very proud we could reach you all. And now the MOOC actually covered all 6 weeks. It's finished. Registration for the certification exam finished on 3rd January too. There is still a chance that you can take the exam until 20th January in case you registered. And good luck for those who are taking that challenge, who had taken the challenge already. If you have tried to get the attendance certificate, it should be available on the Fun MOOC platform soon for you. And now we address the questions that are still open for you. I'm going to read the first questions we got from the community by Akem Fuaf. HIV prevalence is high in key populations all over the globe. How do we target key populations in countries where MSM is illegal? This group of people like MSM, men having sex with men, FSW, female sex workers, or intravenous drug users, etc., might be harboring resistant strains that can, if not detected, lead to a different situation to handle. What techniques can be used to target those key populations?
So we have the reply of Virginie Supervi, who gave one of the lectures. The answer is targeting MSM in countries where same sexual activity is illegal remains indeed a very complex issue. However, there exist ways to reach and target criminalized and stigmatized populations thanks to the work of NGOs, as you might know. In addition, a recent study suggested that the distribution of self-tests may be an additional tool to increase HIV testing among MSM. And she sent us a link. Maybe we can post it.
Yes.
And eventually, the burden of HIV among key populations will not be significantly reduced without strong political will and key population-friendly policies. That will be a necessity for the future. Regarding the resistant strains, if people do not get tested, they will not access treatment, and thus they will not harbor and transmit resistant strains.
Okay, thank you very much, Michaela. Now we address a question that had been asked by Sarah Marzio, and also Michaela will reply to it. What prevents the transmission of SIV from mother to child?
Okay, this refers to one of the first courses where we explained that SIV viruses are very efficiently transmitted among adults, while the rate of transmission from the mother to the child is much lower than in humans. Indeed, the mechanisms and reasons for that are not well known. There are studies ongoing, for example, studies by the group of Ivona Pandrea, and what they have shown is that the expression level of CCR5 on the cells in the blood milk, for example, are lower, so maybe there's less infection in these fluids. The other studies ongoing by Salmi Parma, for example, she shows that maybe the quality of the antibodies or especially the ratio of AGA might be different, and especially in the natural host, like African green monkeys or Sooty mangabees, that might also reduce the transmission rate. And there are finally other studies ongoing by Anshodari, and I think indeed this is a very interesting question, and I hope there will be more studies in the future to understand what is going on. Thank you for the question.
Then we address 2 questions that have been asked in the last chat of the last hangout. Actually, Lativa Berezuga asked those 2 questions. They are we really sure that HIV is untransmissible from a mother under ART, so antiretroviral treatment, to her baby, knowing that the virus remains latent as a provirus in T lymphocytes? Asier is going to give a reply to that question.
Yes, thank you, Latifa, for the question. Yes, of course, we are sure that antiretroviral or the HIV cannot be transmitted from a treated mother to a child, because there are multiple evidence over the years. In presence of the treatment, the transmission of the virus is completely blocked, and the number of infected children has been reduced virtually to zero when the mothers have been treated, or when the mothers are on treatment during the pregnancy and delivery. Also, there is treatment for the newborn, prophylactic treatment that is kept for a few weeks, so that this will avoid any transmission from any remaining infected cell that may come during the delivery, for instance.
Thank you very much, Asier. The other question of Latifa has been, can antiretroviral treatment be targeted according to patients' clinical and biological parameters?
Yes, and partially this is already done. The treatments are adjusted first based on potential resistance of the viruses. So when a person is tested, at least when this is possible to be done very quickly, there are some tests to look for resistance and to adapt the treatment more adequate to control the virus. And in the future, other parameters, there is also actually adjustment of some treatments based on the presence of some high sensitivity genetic factors. For instance, the presence of genetic markers HLA-B57 is not going to allow the treatment with one of the antiretroviral. So there is a screening of the virus, and also sometimes a screening of the host to provide the best treatment possible. And for sure, this kind of personalized approach will be reinforced in the future.
Thank you very much. I hope it's clear now. We got two questions here in the chat that I'm going to address. I'm not sure if we have the answer right now. If not,
I can.
Okay, I asked it and you comment on it, and we can also post a link.
You can complement afterwards.
Online. Okay, so Diego Madias, we address the question you have been just asking. Nice that the two of you here are following us. I'm sorry to address, you said you are sorry to address the question concerning the first weeks of the MOOC, but you would like to know if there are 8 or 9 types of HIV too. So I give the question to our two MOOC directors.
What I would like to say is that regarding the diversity of HIV-1 and HIV-2, what is important is to know that HIV-2 is mostly divided in groups because the diversity between the strains that are known is very, very large. So for example, for HIV-1, you have 4 groups, M, N, O, P, and among each group you have different subtypes, especially in the group M, you have different subtypes and recombinant forms. And in HIV-2, we talk about groups because the diversity is as much as between group M and O, for example. And what is important to know is that some groups are better transmissible or spread more in the population. And for HIV, it's group M, and for HIV-1, it's group M, and for HIV-2, it's group A and B. Whether the numbers now, whether it's 8 or 9, it's not so important, and we will check and post that to be sure, because I think 8 are sure, at least.
Yes, okay, I will post it in the community today. And then we got a last question here of Joachim, if it's still possible to register and get the exam. And 2 more questions here in French. Okay, I have to pass on my French-speaking colleagues here. My French is still very little. So regarding Joachim's question, unfortunately, the registration deadline really finished now. So we had elongated it over Christmas to 3rd of January. But yeah, so it was first to register, the deadline passed, and then the exam started from 7th to 20th January. So unfortunately, the time period is over, but that brings me to a point I wanted to mention towards the end anyways. The chances are very good that we are going to reopen the MOOC on HIV science in 2019 again, for more weeks even, and then you have time to write the exams as well. So maybe some of you were not fast enough covering all weeks, so there is still the chance to complete the MOOC and to write the attendance and the authenticated certificate exams to get the certificates in 2019. The two questions that have been posted?
These are for Aziz.
Okay, they are for Aziz. Okay, maybe Aziz, you can also read the question.
So I will translate the question in English. So I actually will summarize the two comments. So the comments are not directly related to the MOOC. They are related to some results that we published late last year, in December last year. And it's Diego Madias who is asking us to comment about the results a little bit and which are the next steps. So I will do. I will summarize very briefly this in English for the sake of all the audience. So as you know, and this was explained in the MOOC, current antiretroviral treatments are going to block and are very efficient in blocking viral multiplication. But once the virus infects the target cell, the antiretrovirals cannot eliminate these infected cells. And this is the reason why we have to maintain antiretroviral treatment for life, because otherwise the viral replication will resume in almost all people after treatment interruption. And we need to target these infected cells. So what we wanted was to identify characteristics of the infected cells that may allow us to or may help us to identify possible vulnerabilities of the cells. The results that we published a few weeks ago, what are showing is that what we found is that HIV is going, as you know, to infect preferentially lymphocytes. If you have followed the MOOC, you know that it's CD4 T-cell lymphocytes that is the main target for HIV. But we know that HIV cannot infect all the CD4 T cells in the same way. Some of them are resistant, some of them are susceptible to infection. What we found is that HIV chooses or selects CD4 T cells that have a high metabolic activity to infect or to complete the replication cycle. By metabolic activities, we refer to a series of reactions that occur inside the cell and that the cell uses to provide or to obtain energy, and also other factors that are necessary to perform their activity and to survive. And so in this environment, in cells, CD4 T cells that have very high metabolic activity, the virus is going to find all that it needs to multiply. You know, the virus is a very simple particle, so it doesn't have what it takes to multiply and to spread. And in these highly metabolic cells, the virus finds what it's going to need to integrate and then to replicate. With that, we thought that we may have identified a vulnerability, and by blocking some of these metabolic pathways, some of these ways that the cell has to provide energy and other resources, we could block infection and also to eliminate infected cells. And indeed, with some of these molecules that block the metabolism, what we saw in our work is that we can block the first steps of viral replication, in particular reverse transcription. Again, if you have followed the MOOC since the beginning, you know that this is one of the first steps in the viral replication cycle. And not only that, at least in the cells that we infected in vitro, once the cells were infected, if we block the metabolic activity of the cells, we could eliminate these infected cells. Something that we did also with CD4 T cells that we obtained from HIV-infected individuals receiving antiretroviral treatment. In presence of metabolic blockers, we avoided the spread of the virus from the viral reservoirs. Obviously, this is a a very exciting clue, because we now have one characteristic of infected cells that we think that we could target. What is also interesting is that from this metabolic point of view, the HIV infected cell that we have characterized in the laboratory is very similar to, or has some characteristics of cancer cells. And there are different approaches right now being tested in the cancer field to targeting the cellular metabolism, eliminating the cancer cell. So what we want to do is to test the same concept in HIV-infected individuals. Obviously, first we need to identify the best molecules that could be used safely in HIV-infected individuals. Make sure that, because we are targeting a biological activity of the cell, that we will be targeting the infected cells without causing harm to the non-infected cells, because again, these metabolic activities are going to be mobilized or needed by many other cells. And so right now we are testing a panel of molecules. We are testing the effect of these molecules in different cells, not only CD40 cells. We are need to test which will be the effect on tissues, which will be the effect on infected cells in the tissues, something that we don't know. And in parallel, we are trying to get information from colleagues in the cancer field to learn more about the toxicity of the molecules that have been tested already in vivo. Again, this is a first clue. We don't know if this will be applicable in vivo in HIV-infected individuals, but at least it opens a possibility to a new path to target the infected cell, and hopefully we will get new results in a couple of years or 3. At least we will need this amount of time to be ready to test the concept in vivo.
Thank you very much. That was really insights into newest research in that field. Diego, is it clear for you, or is there anything open yet? No comment in the chat, so I think it is clear by now. So we want to finish the MOOC, end it, and say a big thank you to all of you out there who were following our MOOC, dedicated, listening to it, so motivated and interested, posting all the questions. It has been a pleasure to be your community manager. And yes, I hope you liked our MOOC. And it was great having such a big audience from all over the world. So all the best to you. Happy, healthy, and successful New Year 2019 for you. And see you maybe in the next MOOC, HIV Science. Bye-bye.
Bye-bye.